Welcome to Biochemistry DSP channel. In today's topic, I'll be discussing about vitamin A and visual cycle. Vitamin A exists in three forms. It it can exist in aldehyde form as retinol, alcohol form retinol, and acidic form retinoic acid. The retinol, and retinoic acid contains the retinol contains the aldehyde group CHO. Retinol contains the alcoholic group CH2OH. And acid, retinoic acid, will have carboxyl group COOH group in their side chain of beta ion ring. These three forms of vitamin A are physiologically active and are interconvertible. So the provitamin form of vitamin A is beta carotene. This is the structure of vitamin A. So retinol has got alcohol group CH2OH. Retinol has got an aldehyde group in its structure CHO. Retinoic acid has got COOH. And beta carotene is the provitamin form of vitamin A. Oxidative cleavage of this beta carotene can yield two molecules of this vitamin A. It will exist in trans form as well as in the cis form. Okay, cis retinal, or it can exist in trans retinal. Beta carotene will undergo oxidative cleavage to be converted into this vitamin A form, retinal, or retinoic acid. Biosynthesis of vitamin A. So, vitamin A cannot be synthesized by animals. It can be formed through oxidative cleavage of this beta carotenes, that is the provitamin A. Metabolism, when you take this, consider metabolism of vitamin A. In humans, diet contains both plant and animal foods. The dietary retinal ester, okay, the dietary retinal esters will be hydrolyzed by specific, that is, it will be hydrolyzed by unspecific lipid esterase in the intestinal lumen, yielding retinol and free fatty acids. The retinol derived from retinal esters is then absorbed in the intestinal mucosal cells. This absorption of retinol and retinal esters requires bile salt. After absorption from the intestine, a part of this carotene or oxidatively cleaved in the intestinal mucosal cells to yield retinal. Then in the intestinal mucosal cells, the retinol will undergo reduction in the presence of the enzyme retinaldehyde reductase to form retinol. So these two are interconvertible. From retinal you can get retinol, and from retinol you can get retinoic acid. Then the retinol, which is derived from this retinal ester, uh, will be re-esterified with long chain fatty acid in the intestinal mucosal cell to form retinal palmitate. Okay, then it will be released in the blood as a component of chylomicrons. These chylomicrons will transport these retinal esters to the liver, where they will be taken up by the hepatic cells and it will be stored. Okay, 95% of vitamin A will be stored in the liver. Then this from this liver, it can undergo hydrolysis in the presence of the enzyme retinyl esterates yielding free retinol. This free retinol will bind to a protein called retinol binding protein. This is RBP. Okay. So this retinol binding protein with this retinol complex will will transport this retinol from the liver to the extrahepatic tissues, that is the target cells, where it can bind with specific receptors on the cell membranes of target tissues, and it will deliver the retinol. This is how vitamin A will be transported, okay, through retinol binding protein, and very little will be excreted in urine. Functions of vitamin A. So all the forms like retinol, retinal, retinoic acid, all or physiologically active forms, they play a major role in the vision, reproduction, cell differentiation, and growth. It is also required for the maintenance of healthy epithelial mucus secretion. So the main role of vitamin A is it plays a major role in vision. Apart from this growth, reproduction, and maintaining the epithelial cells, a major function or role played by vitamin A is in vision. So this vitamin A, that is retinal, will take part in this visual cycle. Uh, it was discovered by the scientist Wald, hence it can be also called as Wald's visual cycle or rhodopsin cycle, because the light sensitive pigment rhodopsin, which is also called as visual purple. So this visual vitamin A will uh, act as the prosthetic group for this light sensitive pigment rhodopsin, hence it can also be called as rhodopsin cycle. This retina, okay, retina of the eye contains rods and cone cells. Rods are involved in dim light vision, that is night vision, and cones are involved in bright or color vision. This rhodopsin is a conjugated protein which is present in the rods, and it contains 11 cis retinal plus the protein opsin. We'll see what is the role of vitamin A played in visual cycle. So when light falls on the retina, an electrical impulse or signal will be created. The optic nerve carries this electrical signal to the brain. Then the brain processes the signal into an image. So this will happens in the visual cycle. So how it will happen? So when light falls on the retina, immediately the rod cells, which are for dim vision, and cones are responsible for color vision, okay. So when light falls on this retina, this rhodopsin, which is a very sensitive to light, even to dim light it is very sensitive. So when the light falls on retina, the rhodopsin will be exposed to light, and it will be dissociated into opsin and all transretinal. Then this bleaching of this visual pigment will take place, generating a nerve impulse. So in the first step, okay, in the role of vision, it plays two roles. Vitamin A, one that is the one is dissociation of the visual pigment rhodopsin, okay, that event is occurring. Two events will occur. One is the dissociation of the visual pigment rhodopsin. The second event is regeneration of visual pigment rhodopsin, okay. So in the first in the dissociation of the visual pigment rhodopsin, you generate a nerve impulse. So when light falls on retina, what will happen means the retina which contains the rods, okay, which will have a rhodopsin, visual pigment called rhodopsin, which is nothing but it contains 11 cis retinal plus a protein called opsin. So when light falls on this rhodopsin, it dissociates. into opsin plus all trans retinal through a series of events. Okay, so in that series of events, what will happen means, so when a single photon uh, falls on this, so it can immediately excite the cell. So a series of events, that is unstable intermediates, will be produced during this event. That is rhodopsin will be converted into batho-rhodopsin, then to lumi-rhodopsin. It will be converted, lumi-rhodopsin will be converted into meta-rhodopsin 1, meta-rhodopsin 2, and meta-rhodopsin 3. Finally, it will be splits into opsin plus all trans retinal. So all these events will take place uh, within picoseconds to nanoseconds. Okay, so the conversion of rhodopsin to photorhodopsin to batho-rhodopsin will take only picoseconds. Then for this conversion of batho-rhodopsin into lumi-rhodopsin, it takes nanoseconds, and to convert this lumi-rhodopsin to meta-rhodopsin, microseconds and milliseconds will take place. Okay, so it will takes very few picoseconds to milliseconds to convert this rhodopsin into opsin plus all trans retinal. That is the dissociation of this visual pigment occurs. Okay, so this dissociation occurs within picoseconds of illumination, which causes hyperpolarization of the rod cells, which triggers the generation of nerve impulse that is transmitted to the brain through an optic nerve, and the image of the object is perceived. So by which we can perceive this image, whatever image falls on the retina can be perceived because of this production of this nerve impulse. So this cycle was discovered by the scientist Wald in the year 1967, and he has got Nobel Prize also for finding this visual cycle. So when light falls on retina, rhodopsin, which is present in the okay, which is present in the membrane, okay, membrane protein in the retina. So this rhodopsin will be dissociated into opsin plus all trans retinol. Rhodopsin is nothing but it is a conjugated, it contains a conjugated protein opsin plus 11 cis retinal. Okay, when 11 cis retinal plus opsin gives rhodopsin. So when light falls on this rhodopsin, photoisomerization or visual excitation takes place in the rhodopsin, so that rhodopsin will be converted into all trans retinal plus opsin. uh, this okay, so this dissociation, uh, okay, this during this dissociation, what is happening means, this rhodopsin, dissociation of this rhodopsin closes the sodium channels, which in turn causes hyperpolarization of the rod cells. So the closing of sodium channel is brought either by the rapid influx of calcium inside the rod cells, or by lowering the intracellular cyclic GMP level. So this is one event which is occurring during this visual cycle. So dissociation is occurring. The second event is regeneration of the visual pigment rhodopsin. So now this all trans retinal, which is released from dissociation of rhodopsin, is functionally inactive. This all trans retinal is inactive, and it cannot combine with this opsin to regenerate this rhodopsin. So it has to be converted into its active form 11 cis retinal. So only in the active form 11 cis retinal it can combine with this opsin to regenerate rhodopsin. So how this is occurring means, now it is after dissociation to generate a nerve impulse. Rhodopsin, now it splits into all trans retinal plus opsin. And now we are going to regenerate the visual. In the second event is regeneration of the visual pigment rhodopsin. So the all trans retinal, which is inactive form, which cannot combine with this opsin. So what is happening now means, this all trans retinal should be converted into 11 cis retinal. So this all trans retinal in the retinal epithelium, in the with the presence of the enzyme called retinal isomerase, it can be converted into 11 cis retinal, which can combine with opsin to form rhodopsin. One thing, it can this retinal isomerase can convert this all trans retinal into 11 cis retinal, or it can through blood it can enter, this all trans retinal can enter into the liver, where it will be acted upon by alcohol dehydrogenase in the liver, so that this all trans retinal can be converted into all trans retinol. Then this all trans retinal, okay, this for this all trans retinol, again, which is with the help of the enzyme retinol isomerase, which is present in the liver. Okay, so with the help of retinol isomerase in the liver, it can be converted into 11 cis retinol. Then again, this 11 cis retinol, with the help of the same enzyme alcohol dehydrogenase, it can be converted into 11 cis retinal. Through blood it can enter into the retina, where it can combine with opsin to regenerate this rhodopsin. So this is how it will regenerate. So a continuous supply of 11 cis retinal is required in the rod cell to maintain the visual cycle. So in visual cycle, what are the events is taking place means, one is the dissociation of the visual pigment rhodopsin, which is the first event. Why this event is necessary means to create a nerve impulse, so that the nerve impulse can be transmitted to brain through optic nerve, and the image of the object can be perceived. Okay, so during this first event, what is happening means, that during the dissociation of the rhodopsin, it will close the sodium channels, which causes hyperpolarization of the rod cells. So how this closing of sodium channel is brought about means, it can be brought about by rapid influx of calcium ions inside the rod cell, or by lowering the intracellular cyclic GMP level. So in this rod cell, cyclic GMP, okay, cyclic GMP will be acting as a second messenger to regulate the opening and closing of this sodium channels. In dark, increased intracellular cyclic GMP level. So in dark, what happens means, increased intracellular cyclic GMP level will allow the opening of sodium channels, whereas in the presence of light, a lowering of intracellular cyclic GMP level closes the sodium channel. So by closing and opening the sodium channels, it will regulate. Okay, the cyclic GMP will act as a regulator in closing the sodium and opening the sodium channels. So in the dark, increased intracellular cyclic GMP allows the opening of sodium channel, whereas in the light, lowered intracellular cyclic GMP level closes the sodium channel. So the role of protein, transducin protein, okay, this transducin protein has been suggested to regulate this cyclic GMP dependent opening and closing of sodium channels. So this protein is very important to uh, activate this cyclic GMP to open up or close the sodium channel. When rod cells are exposed to light, this transducin GTP will activate phosphodiesterase enzyme through a cascade mechanism. So the activated phosphodiesterase will hydrolyze the cyclic GMP, causing lowering of intracellular cyclic GMP level, which will close the sodium channels, resulting in hyperpolarization of rod cells. So this is the first event which takes place during the generation of nerve impulse. So then the second event is regeneration of 11 cis retinal. So this all trans retinal, which is released from dissociation of rhodopsin, which is inactive, which cannot combine with opsin, so it has to be regenerated. So it has to be converted into an active form 11 cis retinal, so that it will regenerate rhodopsin. So how it can be done means, with the help of the isomerase enzyme retinal isomerase in the rod cells, it can be converted, it can be isomerized into 11 cis retinal, by the enzyme which is present in the retina, that is retinal isomerase, or it can be enter into liver, where it can enter into liver, where it can be acted upon by the enzyme alcohol dehydrogenase, where it can be converted into 11 cis retinal, so that it can combine with opsin to form rhodopsin. So this is how it will maintain a continuous supply of 11 cis retinal to maintain visual cycle. Then what is meant by dark adaptation time? Bright light, okay, suddenly when a bright light falls on rhodopsin, it bleaches, okay, that is it converts rhodopsin into opsin plus alternates retinal. So this event, the dissociation of visual purple rhodopsin, is called as bleaching of visual pigment or photo bleaching. So bright light will depletes the rhodopsin. Photo bleaching, that is the sudden shift from bright light to darkness causes difficulty in seeing. So when that okay, that the time interval to adapt this sudden shift of bright light to darkness is called as dark adaptation time. So this time increases during vitamin A deficiency. For those who have vitamin A deficiency, this dark adaptation time also will increase. So rhodopsin is synthesized within a few minutes and vision is improved in the dark. Okay, so to synthesize this rhodopsin, it takes few minutes so that vision can be improved in the dark. But for vitamin A deficient patients, rhodopsin cannot be synthesized within few minutes, so it will take a longer time to regenerate the rhodopsin. Hence, their dark adaptation time also will increases. So, adapting to dark environment is called as dark adaptation time. So, the time which is required to synthesize rhodopsin in the dark is called as dark adaptation time. It increases during vitamin A deficiency. Then, what are the other biochemical functions of vitamin A? Means, other than vision, it plays a major role in growth and cell differentiation. So, in the form of retinol and retinoic acid, which acts like steroid hormones, and they will play an important role in the cell growth and differentiation. So, it will bind with specific receptors which will interact with the DNA and regulates the expression of specific genes, resulting in the synthesis of specific proteins, and they will regulate the cell growth and differentiation. And vitamin A is also necessary for health, maintaining a healthy epithelium and mucus secretion. So it is required in the variety of organs and tissues like respiratory and urinary tracts, eyes, tongue, and mucus secreting glands, skin. Okay, where this retinol and retinoic acid is are used to maintain the healthy epithelial structures by preventing the keratinization of epithelial cells. So it helps to maintain a healthy epithelium and mucus secretion, and it has got a very good antioxidant property. This uh, antioxidant property is mainly due to the presence of beta carotene. Beta carotene acts as an antioxidant. Antioxidant means it inactivates the free radicals and protects the tissues from their toxic effects. These free radicals are strong oxidants and are highly reactive, so they can damage the biomembranes by causing lipid peroxidation. So the role of free radicals can create cancer or chronic diseases and aging process. So vitamin A acts as a natural antioxidant to cleave these free radicals and decrease the risk of cancer and other chronic disease like coronary heart disease, cataract formations. That is also one of the important function of vitamin A. Then other functions includes it plays a major role in improving the immune system. So those patients who have less amount of vitamin A may be prone to infections. Then it is also required for the synthesis of transferrin, ion transport protein. So during its deficiency, it may lead to iron deficiency anemia. So these are the some of the functions of vitamin A, other than role, and it's play a role in vision. Other than its role in vision, it has got other functions also. Then deficiency manifestations of vitamin A. So the main manifestation is night blindness or nyctalopia. In night blindness, the patients fail to see the object in dim or poor light, as the rod cells become less sensitive to light due to lack of the visual pigment rhodopsin. So in the case of night blindness, the dark adaptation time increases, and prolonged deficiency of this vitamin A may cause irreversible damages in the rod cells, that is the visual cells. Okay, so the visual acuity is diminished in dark light. So the patient cannot read or drive a car in poor light. Their dark adaptation time is also increased. Next is xerophthalmia. The conjunctiva of the eyes becomes dry, thick, and wrinkled. That condition is called as xerophthalmia. So the lacrimal glands, okay, the lacrimal glands degenerate and stop to produce tears, which causes dryness of the cornea and conjunctiva. This clinical condition is said to be dry eyes, okay, or xerophthalmia. Xerophthalmia means dry eyes. So, if the condition prolongs for a longer time, it may lead to keratinization and ulceration of the cornea. And if untreated, it may produce opaque. Totally, the cornea becomes opaque, resulting in permanent loss of vision, that is blindness. That condition is called as Keratomalacia. Then Bitot spot. Okay, these are white opaque spots which develop in both the sides of the conjunctiva due to vitamin A deficiencies. These are grayish white triangular opaque spots which are present nearer to the conjunctiva of the eyes. When xerophthalmia condition, okay, when xerophthalmia condition persists for longer time, it may progress to another advanced condition called as keratomalacia, which will lead to permanent loss of vision. In the case of severe deficiency, it will lead to complete blindness. And other deficiency symptoms is anemia. It will lead to hyperchromic and normocytic anemia, which is due to vitamin A deficiency due to impaired iron transport. Then decreased resistance to infections. Vitamin A deficiency also lowers the body resistance to infections due to impaired differentiation of cells of immune system. Then poor growth and functionally impaired reproductive system. So it will lead to uh, poor growth of bones, nervous system, reproductive system, as well as teeth. Male rats are shown to defective spermatogenesis, and female rats fail to carry the pregnancy to full term. Hence, it is vitamin A is also required for cell differentiation in spermatogenesis. So these are the deficiency manifestations of vitamin A. What are the causes for this vitamin A deficiency? Is it may be due to decreased intake of vitamin A or obstructive jaundice, which may cause defective absorption of this vitamin A, since it is a fat soluble vitamin. Vitamin A is a fat soluble vitamin, so it requires bile salts for its absorption. Due to obstructive jaundice, there is defective absorption of vitamin A, and cirrhosis of liver. Since vitamin A is stored in the liver, cirrhosis of liver can also reduce the synthesis of retinol binding protein. And severe malnutrition, where amino acids are not available for retinol binding protein synthesis. Chronic nephrosis, where retinol binding protein is excreted through urine. So all these factors may leads to vitamin A deficiency. The rich sources of vitamin A is cod liver oil, shark liver oil, then liver, kidney, egg yolk, milk, cheese, butter. All these are good sources for animal sources. And in the case of vegetable sources, carrot, papaya, pumpkin, mango, spinach, all these are richest source of vitamin A. RDA, recommended dietary allowance. Okay, so this recommended dietary allowance per day of vitamin A in children includes 400 to 600 micrograms per day. For men, 750 to 1000 micrograms per day. For women, it is 750 micrograms per day. And during pregnancy and lactation period, 1000 micrograms per day is required. It can be represented in the form of IU per IU also, that is 5000 in adult, 5000 IU of vitamin A is required, and for men it is 5000 IU, or for women it is 4000 IU per day is required. One IU corresponds to zero point 0.3 microgram retinol. One retinol equivalent, that is RE. Okay, in that way also you can represent vitamin A. One retinol equivalent is equal to 1 microgram of retinol or 6 microgram of beta carotene. So in that way also you can represent this vitamin A. Toxicity or hypervitaminosis A. Since all fat soluble vitamins are stored either in liver and adipose tissues, it may lead to hypervitaminosis or toxicity. Since this vitamin A is also fat soluble vitamin, it can be stored. Excess amount can be stored in the liver, where it may lead to toxicity problems like anorexia, irritability, headache, drowsiness, bone and muscle cramps, loss of appetite, skin disorders, that is dry and rough skin, hair loss, alopsia, increased liver size in children, vomiting and diarrhea, congenital malformation in the growing fetus, spontaneous abortion, and birth defects. All these are due to hypervitaminosis A. This hypervitaminosis condition is predominant in the case of Eskimos who eat polar bear liver, which is extremely rich in vitamin A. It contains, this polar bear liver contains about 6000 to 20,000 IU per gram. So it may also leads to toxicity of vitamin A or hypervitaminosis A. So hypervitaminosis means increased level of vitamin A in the serum. This hypervitaminosis A is seen in Eskimos. Mainly it is in Eskimos who eat polar bear liver. This liver is richest source, so it may leads to toxicity problems. Thank you for watching this video. Subscribe and support the channel Biochemistry DSP.